Hello, my name is Catherine Lowry, and alongside the work of Krenna Rahr, Alexa Kiblinger, Griffin Howell, and with the faculty advising of Dr. Michelle McWhorter and Dr. Gwen Davis, I completed summer research in summer 2025 on the impacts of food insecurity on gene expression in C57BL6 mice. Food insecurity is a worldwide issue affecting 1 in 11 people globally, leading to malnutrition, increased rates of illness, and has been shown to increase the potential of mental health issues. In this study, C57BL6 mice, or lab mice, were treated to feast-famine paradigms for 3 weeks and sacrificed to create cDNA for quantitative polymerase chain reaction, or qPCR analysis. This was used to measure the relative gene expression in DAT, DRD2, and DRD1, which are the genes of interest in this study. Analysis was done on 2 cohorts, one without a recovery period and one with. For both cohorts, the dorsal medial striatum and dorsal lateral striatum were analyzed. Ultimately, the hypothesis was validated with food insecurity epigenetically changing the expression in the nigrostriatal pathway in mice when comparing the secure and insecure groups. Food insecurity is defined as the limited or uncertain availability of nutritionally adequate and safe foods. And as previously stated, food insecurity is associated with elevated risk of mental illness and disorder and has been related to increased problems with working memory and executive function. In this study, we focused on how food insecurity affects the dopaminergic signaling essential for motivation, goal-directed behavior, cognition, memory, and impulse control. It was hypothesized that mice undergoing food insecurity paradigms, specifically without a recovery period, would be affected in their dorsal medial striatum and dorsal lateral striatum within the DAT, DRD1, and DRD2 genes, which are our genes of interest again. For the study, we used mice brains as they are great counterparts to human brains, even if they are roughly 1,000 times smaller in volume. They have the same structures and functions, just on a more adaptable and ethical scale for science versus a human brain. In specific, we looked at the dorsal striatum, which is a key brain structure involved in movement control, skill learning, and habit formation. The entire dorsal striatum was separated into the dorsal lateral striatum and the dorsal medial striatum to get different points. Mice were divided into food secure and food insecure groups. Baseline weights and food consumption were taken over a course of 4 days. After these 4 days, mice were put under the feast-famine paradigm, which you can see in Table 1. In mice with the recovery period, so the recovery period cohort, this was followed by 9 days of 100% of food before they were sacrificed and brains were rapidly dissected out, sliced into 1 millimeter increments using a brain matrix on ice. These slices were moved into RNA later buffer, where 0.75 millimeter punches of the tissue were made to remove the DMS and DLS separately. You can see these in Figure 1, the images. Tissue punches were then placed into microcentrifuge tubes containing RNA later. This was done for each cohort of mice. For cDNA synthesis, for qPCR analysis, it was done using the tissue punches from the RNA later from the mice, and then they were used for the qPCR analysis in a user-defined protocol using the CF96 Maestro, which you can see the protocol in Table 2. These qPCR reactions were done using a master mix of ITAC Universal Cybergreen Supermix, RNase-free water, both forward and reverse working nucleotide primers. You can see an illustration of this master mix in Figure 2. These reactions were done on a 96 well plate with 19 microliters of master mix to every 1 microliter of cDNA in every well. In all groups that were tested, there was a decrease in gene expression for each of the genes of interest in the food insecure group. In Figure 3, you can see the normalized expression in the dorsal lateral striatum in the 3 genes of interest, and in Figure 4, you can see the normalized expression in the dorsal medial striatum in the 3 genes of interest. As the dopaminergic system is heavily involved in motor movement, attention, and impulse control, this data gives us a biochemical backing to show the suggestion of higher increase in diagnosis, including those symptoms, could be related to food insecurity in the United States and globally, when backed by behavioral analysis data to show mental illness or disorder. Another cohort of mice would be beneficial in future work to show behaviorally and genetically the argument for this. Ultimately, this study showed that food insecurity is epigenetically changing expression in the nigrostriatal pathway in mice, and there is slight evidence to suggest that this epigenetic change could be reversed through food security in mice. And we care because food insecurity has been shown to increase problems within working memory and deficits in executive function in humans, and being able to see this as well in mice is very prevalent in today's psychological state. Thank you, and if you have any questions, please feel free to reach out to me, and I would love to get back to you. Have a great day.